sadece match'lere bakarak.
No, the root mean square distance is actually, it uses the three-dimensional coordinates of the points. To compute these distances, we need to know the structures of proteins. You cannot, just knowing the primary structure, which is the sequence.
Which mesh do you choose as point A, B, C, and D?
Yeah, that's the alignment problem. Which points are going to be? Exactly. Yes. What you can do is this. That's going to be really interesting. You can do sequence alignment on these structures and get the matches from the sequence alignment. Okay, your matches can be provided by your sequence alignment because sequence alignment is going to provide you a match for you, right? A matching amino acids are going to be provided by the sequence alignment. Now, you may assume if you really trust your sequence alignment, saying that these are the actual homolog, match, these amino acids should be the ones that should be matched, you can take it as your structural alignment and see how these matched amino acids are similar in shape. You can do it. Doing sequence alignment first, and whether actually that I may even your last assignment may be something like this. I may provide the code. No, you're not going to able to. It's going to be very easy if I do that. Because I'll provide you the code that this ON time code that does the superimposition. All you need to do is run sequence alignment, your assignment first, without any modification, no flanking. So run assignment 1. You run a local alignment, local sequence alignment on 2 proteins, and after you get which amino acids are matched, run this other program considering these matched amino acids and the three-dimensional structures to see whether the matching provided by the sequence alignment actually shows that these 2 structures are similar or not. So why don't other people do that? Is that sequence alignments, sometimes structure is conserved better than sequence. In other words, you can have, in terms of sequence, very dissimilar proteins, only 10% sequence similarity. However, their structures may be really similar. So the sequence alignments may give you incorrect matches. By just looking at the sequence alignment, you may not even get a good sequence alignment because the sequences may be completely different. Only 7% or 10% identity. So in that case, the amino acid matches you are going to get are going to be very unreliable because the sequence information does not show you anything about similarities. Their sequences are not similar. Now in that case, what you need to do is you need to find the similarity in 3D. So you need to find which amino acids should be matched with each other by making use of their three-dimensional coordinate information. That is when we really need structural alignment. I mentioned some of these things. There can be structure alignment, can be sequence order dependent or independent. And our goal will be, given 2 structures like this, whether they're similar or not, to find out first the matching amino acids and then superimpose them. Which matching is going to give us a better superimposition. And this, when we are superimposing these amino acids, these 2 proteins, we ignore the unmatched parts. The unmatched parts are not taken into account when computing this RMSD. We only consider the matching parts. And structural alignment, in general, is local. There may be local substructures, local shapes that are similar. Overall, the proteins may not be similar, but there may be some local shapes that are similar. So when we are superimposing 2 proteins, we superimpose only these local matching shapes. The non-similar parts are not tried to be put on top of each other because they are not similar to each other in terms of shape. Here, the optimal alignment tries to solve 2 objective functions and try to optimize 2 things. that are actually conflicting with each other. Okay, so here our goal in structural alignment, given two protein structures, find the maximum number of amino acid matches between them, such that when we superimpose these matched residues, their RMSDs are going to be minimum. Why are these two approaches conflicting? Let me tell you one extreme case. I can give you, I can provide you structural alignment between two protein structures, actually between any two protein structures. Give me completely non-similar shapes. I can give you a matching which is going to get you RMSD of 0. Okay? I mean, if your goal is to have RMSD 0, minimize RMSD, I can say that only the first and second amino acids are matched to the first and second amino acids of the second protein. Okay? I mean, matching 2 points only. I mean, they're just lines. These shapes that I tell you similar are just simple line segments. And the bond length between, the length between consecutive carbon alpha atoms are same in any protein. So this line segment is going to be 100% similar to the other line segment in the other protein structure. Okay, I found a similar shape, which is a line segment. Okay, I mean, which gives you an RMSD of 0, but biologist is going to say that this line segment is something biologically uninteresting. Okay, this biological shape, just these two amino acids, the shape that is formed by these two amino acids is not interesting at all. So what do? You increase these a match of 2 is not interesting. So we try to increase the number of matching amino acids, which is going to increase your RMSD. So you may have found that, for example, so these are conflicting objectives. As you put new matching residues into this function, this RMSD, you are going to be potentially increasing root mean square distance. So here, therefore, that's the main thing most algorithms differ. When you look at solution of different structural alignment algorithms, one algorithm says that I have found a match of 30 amino acids with RMSD 3. Okay, so one algorithm may say that I have found a match of 30 amino acids between two proteins. 30 amino acids may also be interesting biologically. It may be a structural motif, for example. And RMSD is 3 angstroms. So by the way, the unit of RMSD is angstroms. Here, distance is measured in angstroms squares. We take the square root of these angstroms, and the final is going to be is in angstroms. Okay, one algorithm may tell you that I have found a match of 30 amino acids between these two structures, and the RMSD between them is 3 angstroms. And another tool may be saying that I have found a bigger match. I have found in my alignment, I have 50 amino acids that matched. So this algorithm is telling you that there is a larger part of these two structures that are similar, but my RMSD is 4 angstroms. Okay, so which solution is better? And the answer to that is, I mean, you can say there's no solution that is better. I mean, there's no universally accepted solution to that. I mean, this is a smaller shape, which is more similar because the RMSD is smaller. This provides you a larger part of the same protein structures. I mean, these results can be taken for the same two protein structures A and B, not different protein structure. For the same two protein structures A and B, if you run one algorithm, one structural alignment algorithm, that algorithm gives you this result, another algorithm gives you this result. And which one is better? I mean, it's just a preference. It's very subjective. So that's what is difficult. I mean, okay, some tools, they try to find small, very similar structural motifs shared between two proteins. Some other tools try to match larger parts of these two protein structures at the expense of a little higher RMSD. So in terms of, if you look at this larger structure, there are going to be more dissimilarities between them compared to this two. This is because it's a smaller structure. They're more similar to each other. But adding new amino acids, you're potentially increasing this RMSD. And where to stop is really, there are no universally accepted measures in this thing. So even the problem definition is difficult. Okay. But usually there are certain things. For example, an RMSD of 7 is really bad. Okay, like 5, 6. So we want similar shapes usually have RMSDs around 3 or 4. We don't want to go higher than 4. So there are some universally accepted intuitions like this. If you say that I have found two similar structures, 120 residues matching, RMSD is 10. You shouldn't be happy about this. RMSD 10 is not a very good thing. Okay, so RMSD of 10 is considered not very good. So there are some universally accepted, so 3, 4, maybe 4.5. Those things can be considered okay alignments. But if you compare these two, I mean, everybody, if you ask this to many different structural alignment experts, you can get different answers from each of them. I mean, you can get, maybe they may be split 50-50, I don't know. This is really a hard thing to decide which alignment is better than the other. Yes? Yes, you can do it. I mean, if you want larger alignments, if your goal is to find more parts of the structures that are similar, you may give more importance to the length and less importance to RMSD, but still you may have certain thresholds saying that do not pass this RMSD. 5 angstrom is the maximum RMSD I can accept. You may have something like this. So it's really an interactive process. You try different algorithms may give you different things, and you're trying to find a balance between these two conflicting objectives. Some people have tried to combine these two to get a single measure, like this. For example, length divided by RMSD. If you do this, for example, because the smaller the RMSD, the better the score. The larger the length, the better the score. You can put these two together to get a single score. In that case, this is going to have 30 divided by 3, 10, and this is going to be 50 divided by 4, something greater than 10. Okay, so you are going, if your scoring function is this, you are going to prefer this better, but you may argue whether this is a good scoring function or not. Okay. What about this? If I do this, maybe it's going to, it's not going to change if you compare them, but I mean, there's no universally accepted scoring function like this. This is what I'm trying to say. Now let's talk about structural alignment algorithms, what we are trying to find. Similar to sequence alignment, the result that they are going to provide us is going to be similar to what sequence alignment provides us. It's going to provide correspondence between amino acids that you can do sequence, that you can do with sequence alignment. But instead of using the amino acid letters, we are going to ignore the amino acid types completely and just, most algorithms just use structural information. They are three-dimensional information when they are finding this matching. And because of examples like this, we need structural alignment for aligning sequences like this. They share, these are myoglobin and beta hemoglobin, the alignment result of them. Their sequence, this is a wrong sentence, by the way. Sequence homology is 85%. This is an incorrect use of the term homology, okay? Homology is binary. There's either homolog or not. It's not 85%, 8.5% sequence. It should have been their sequence identity or sequence similarity is only 8.5%. So if you try to look at the sequences, they share only 8.5% similar amino acid types, same amino acid types, but the structural alignment reveals a much higher structural similarity. Okay, so superimposition, I told you that it's can be done very efficiently by a linear time algorithm, but find the best translation and rotation to superimpose two structures is no. And I talked about the root mean square deviation, which is computed like this. So it's the same formula there. And the root mean square distance between these two protein structures are shown here. And as you can see, as the protein is here right now, this is not the minimum RMSD we are showing here, we are computing. For example, you can get the RMSD smaller by positioning this green protein down. Okay? If you take it down, RMSD is going to decrease. So obviously this positioning does not show the minimum RMSD. But how are you going to position the red? How would you position it to get minimum RMSD? For example, because they're not exactly same shapes. So where is this 1 going to be? If this 1 and 5, they're exactly on top of each other. This 3, so it's just tries to minimize this average distance. So not every amino acid can be on top of its matched counterparts. So they can be in different places. And I also told you that the unit is angstroms. And if we have exactly identical shapes, the RMSD we are going to find is going to be 0. We can exactly put each matched point on top of its corresponding. And similar structures, usually the RMSD is between 1 and 3. And distance structures, RMSD is greater than 3 angstroms. Still 4.5, 4 or 5, up to 5 can be an accepted similarity if you have a large protein structure, of course. If you have RMSD of 5 between 10 amino acids, that's a bad thing. So if you're just matching small parts of your structure, you are looking for RMSD between 1 and 3 angstroms. And there are different variations of this measure RMSD. Here, the disadvantages of this original definition of RMSD is that we treat all atoms equally. For example, maybe if we know that certain residues are on the surface, and it's known actually, we are comparing two structures, we do not give different values to these distances, whether they're on the surface or whether they're at the core. Maybe we may allow more deviation at the loop regions maybe, or at the terminal parts of these proteins. We do not care whether they're at the terminal or whether they're at certain critical regions. All amino acids here are treated equally when we are computing them. There are variations of this, like weighted RMSD, or ARMSD, BRMSD. There are different variations of RMSD that you may encounter in papers, which give different weights to different parts. And sometimes the best alignment does not always mean minimal RMSD, meaning that functionally, biologically, true alignment may consider other things as well. And recent years, in the last five years, there are also techniques which tries to align proteins considering small, by considering them not as rigid bodies, but allowing certain, by treating them as flexible objects. For example, a protein here, when we compare it to another protein, which is in another conformation like this, maybe the protein structure we have is in this shape because it's interacting with a certain compound. Maybe we may consider this as structurally similar to that if maybe this protein sometimes, so proteins are not rigid bodies in general. When they are interacting with compounds, they change conformations. There are small conformational changes. For example, things such that may be observed in proteins. They are not fixed static shapes. When we solve them with X-ray crystallography, we make them static, but in their native environment, they go over conformational changes. So they are flexible. Some parts of them are flexible. And recently, in the last, this is just, I'm telling you for information, there are certain new structural alignment approaches which takes into account these flexible regions and tries to align proteins considering the flexibility. And here are the alternative RMSD measures: ARMSD, BRMSD, WRMSD, and these were from a paper 12 years ago. And there are many, many different structural alignment algorithms. Here are a list of some popular of them. Even, I wrote a structural alignment program during my PhD. I didn't list mine here. It's not one of the popular ones. But it's based on some computational geometric measures from proteins. What I did was I was measuring the curvature and torsion angles locally at each structural shapes, and instead of and I used the dynamic programming sequence alignment type approach to align these local geometry, similar local geometries. So similar curvature and torsions were aligned with similar curvature and torsions in the other protein. But mine is not listed here, as I said, because it doesn't provide very good results. It can provide some small structural motifs, very good, but local similarities may not always lead you to a global similarity. That was the disadvantage of the technique I proposed. But there are different methods, as you can see. These are classified based on whether they're distance-based. Distance-based methods, they use the interatomic distances or inter-amino acid distances between, or intra-amino acid distances between proteins within a protein. And there are vector-based methods which represent a protein structure as a set of vectors, because our secondary structure elements are a helix, for example, is a vector. A helix has a direction, and this can be treated as a vector. So they abstract the protein structure as secondary structure elements. And in the context of this course, I'm going to talk about in detail about the structural algorithm. It's an iterative dynamic programming algorithm. And I'm going to talk about DALI, not in many detail, but just the general, the main steps of the DALI algorithm. And I'm not going to talk about the others, just I mentioned them here. If you're interested in, you can read the papers about them. Here, the structural algorithm I'm going to talk about in detail. It uses dynamic programming to solve the structural alignment problem. But since single dynamic programming is not going to give us the optimal solution, it does this iteratively. It iteratively runs a dynamic programming algorithm to find the optimal, at the end, to find a good match between the sequences. And it doesn't guarantee an optimal solution. None of these techniques guarantee an optimal solution. As I said, even the notion of optimal is not a universally accepted notion. Now, so we don't have a globally accepted objective function. That's why each of these structural alignment tools give you different results. Now, here it starts the structural algorithm. It starts with an arbitrary alignment. Actually, the first step can be made better, more intelligent. Instead of starting an arbitrary alignment, start with sequence alignment, for example. Start with what sequence alignment gives you as matching amino acids. But, it can also start with an arbitrary alignment. Just random alignment of amino acids. Just randomly align amino acids with each other between these two proteins. It starts with this random alignment, and in each iteration, it tries to make this alignment matching better. It changes some of the matches like this. So the first step, if we are given an arbitrary alignment between these two proteins, the second step can be done in linear time, in one time. If we know the matching amino acids in part B, we can superimpose these two shapes to get minimum RMSD. Now, in other words, it's like this. Start with a random alignment. Just try to bring these two proteins together, close to each other, but by using a random alignment, which may not make sense at that time. Just try to bring them closer in each other in 3D space. That's what step 2 does. And after that, we compute a new alignment, a new matching of amino acids by running dynamic programming on something different, not on sequences, on the 3-dimensional positions after the superimposition. It's like this. We compute a scoring matrix. At step 3, what we do is, in sequence alignment, what do we need? What we need is, what is the score or penalty of matching this amino acid with this amino acid? What is our, we have this blossom or PAM matrices, which tells us if you match this type of amino acid with this type of amino acid, this is the score you get, or this is the mismatch penalty you get. This is what we need in sequence alignment, right? So step 3 does the same thing, but it gives us a structural alignment matrix. It's same as scoring matrix, which tells us what is the score of aligning an amino acid here with another amino acid in the other protein. And it does this, since it's a matrix, so let me actually do it like this. So I have a certain protein here, and I have another protein here. But I just have one, in the first step, I do a random matching, okay? This one is matched with this one is matched with this, okay? Just a matching of 2. The size of, the step 1 does not detail how many, if their lengths are different, how are you going to provide this random matching? What is the length of the, what is the number of matched residues? It doesn't give the details of that. But you can just pick 3 amino acids or 10 amino acids randomly to do this matching. But imagine this is my match. And at the end, after the superimposition, I can take these 2 points together. They are on top of each other, or they are as close as possible, or they may be exactly on top of each other. If you choose 2 of them, you are going to get an RMSD of 0 at step 2. If you just 2 points, if you just, actually, no, you may not get RMSD of 2. I mean, the length between the chosen points, they may be different if they're far from each other like this. Anyway, the first step may give you a ridiculous alignment like this, or ridiculous superimposition like this. Okay, it's not meaningful. The matching is not known. Now, at the third step, what we do is, by considering each amino acid in each protein, by considering each amino acid in each protein, we compute the score of aligning this with this, with this, with this, with all of them. Okay, for example, we have an M by M matrix, so N and M, the number of amino acids in respective protein structures. And here an entry ij shows the score of aligning the ith amino acid with the jth amino acid. And this score is completely is computed only based on their distance in the superimposed structures. So it's like we are going to get a higher score by matching close by amino acids, and we are going to get a bad score by matching far away amino acids. So closer amino acids in this, so second step brings these 2 proteins together, and closer amino acids are going to get a higher score to match, and far away amino acids are going to get a bad score if you match them. So we are going to get mismatch penalty if we are computing a BLOSUM PEM type of matrix based solely on their three-dimensional positions of these amino acids. So this is what we do in step 3. So this step 3 is not dynamic programming at all. Okay? So this step 3, by just looking at distances, just finds out whether it's a good idea to match them or it's a bad idea to match them. Okay? If they're close, it's going to tell us that it's a good idea to match them. If they're far away, it tells us it's a bad idea to match them. So the third step is the construction of our BLOSUM matrix, similar to, so it's not dynamic programming. But if we have such a matrix, what can we do? We can run dynamic programming to find the best matching of amino acids. So this matrix is going to tell you what is a good idea to match which amino acid, which amino acids are likely to be matched. And by using this matrix, you can run dynamic programming. Now you can run the same algorithm as sequence alignment, but do not use the original BLOSUM matrix, but use this matrix to find this match-mismatch scores. Your match-mismatch scores are on this matrix. You run your dynamic programming algorithm, and at each time, for example, when you're filling in the ijth entry, you are going to have gap penalties again, and you are going to have match or mismatch score. The match-mismatch score you are going to use here, the score of ij in the dynamic programming algorithm, you are going to get this value by a lookup to this table. Whether matching inj is a good thing or bad thing, you are going to find it from this table. Okay, so what will happen is that after step 4, so here it says that gap cost is 0. We can also use some negative number if you want that, so it's a parameter. So after step 4, what we have at the end is a new set of matching residues. We hope that we had initially random, ridiculous matching residues, this one and this one. After step 4, we see that, oh, okay, these amino acids are also close to each other. Let's match them as well. Or maybe after this superimposition, we see that, maybe we may see that the ones that were matched initially, now it's actually closer to something else, somebody else, and we may even change our decisions that we made in the first step. There may be new additions, new matched residues, and we may also change some of our earlier decisions. So step 4, the result of step 4 is another set of correspondences between amino acids. So what do? We have new set of correspondences. Run step 2, 3, 4 again. Okay, so after we have these new matching set of amino acids, we may re-superimpose them. We may, I have this new thing, I may change I may superimpose them again based on these new matches. After I superimpose it again, its three-dimensional position is going to change, so this matrix is going to change as well. After this matrix is going to change, you are going to run the dynamic programming algorithm again. Again, you may get a new set of matching residues. You continue this iteratively. That's why it's called iterative dynamic programming. You iterate these steps 2 to 4 until the overall score or the number of matching amino acids do not change in the next iteration. If you find that amino acids do not change, they are not going to change anyway. I mean, if you continue how many iterations, if they get to a fixed point, they are going to stay at that fixed point, because if your superimposition does not change, this matrix is not going to change, this matrix is not going to change. So you are going to get stuck at a certain set of matching amino acids. Now, here you can immediately see that this arbitrary alignment at the first step is really important. I mean, our final result is very much dependent on our initial alignment. If you have a really bad initial alignment, you can stuck at a very bad solution. Or if you have, the result is really dependent on your arbitrary alignment initial that you use in step 1. and therefore we added the step 6 at the end. Repeat this for different initial alignments to see which one provides you a good answer. And as I said, an intelligent mechanism may be to use sequence alignment. If your sequences share like 30% similarity, maybe you can use the sequence alignment result as your initial alignment. Okay, so is this algorithm clear? Now here's an example run of the algorithm from the original paper that introduced this algorithm. Here we have a protein of size 5, another protein of size 7, right? Yeah, I have size 5 and size 7. These are two proteins that are going to be matched. And as we can see, I mean, you can actually manually align them. You can just see that here this A, B, C, D, E matches with capital A, B. So, sorry, the quality of the image is really bad. Let me use the board to show you in more detail. So, one structure is like this, A, B, C, D, E, the lowercase letters. And the other structure is A, B, C, D, E, F, and G, I guess. Without, manually, so this is one protein. And this is the other protein. So manually, you can immediately see the optimal alignment is this. BB, then CE, DF, EG. Okay, this is the optimal alignment. A is matched with A, B is matched with B, C is matched with this E here, D is with F, and E is with G. So matching these 5 with these 5. So I have some linear shape here. That linear shape is observed. This is the linear shape I have here. So this C and D should not be matched to anything. So matching is between A, B, E, F, G, capital A, B, E, F, G in the other sequence. So this is what I should find at the end. Now the struct algorithm works like this. Initially, this is an arbitrary equivalence which matches C, D, E, F, G, capital C, D, E, F, G with A, B, C, D, E. And this superimposition gives us something like this. As you can see, the matching parts, they are not totally matching, and this is the best superimposition provided by this arbitrary matching. And this superimposition now puts these two proteins close to each other, and based on their positions, we compute a score matrix. And this is not the dynamic programming matrix. And this score just based on how close these amino acids are in 3D space. For example, look at this one. We initially matched this with this maybe, okay, but here its position indicates that you can also match it with this one, okay? So if you can follow the cursor, there's this hand here, this residue in the white protein, maybe it was matched to this one initially, but it can also be matched to this one. So because it's positionally close. Also this one can be matched to this one or this one. So this superimposition gives us new alternatives about these matchings. It's going to give us new alternatives, and these new and the best way, by looking at these positions, we find a new set of the dynamic programming based on this scoring function we have. It's going to give us the optimal way of aligning these two shapes. It's going to give us new correspondences, and these new correspondences may be A, B, C, C, E, D, E, DF and EG, and it's like this. It tries to match these with these, and this is the superimposition we get. And at the end, again, we see that these middle black points here, they can either be matched with this one or this one can be matched with this one or this one. And the dynamic programming is going to give us the best way to do this. But when doing the dynamic programming does not know the new superimposition. It cannot see that. If you match it with this one, you can also get this superimposition better. It doesn't see that dynamic programming. It just runs on this scoring matrix, or your score matrix, which is computed based on the distances. And we hopefully get the optimal structural alignment at the end. So iteratively, at each iteration, the number of matched residues change because your superimposition change. After that point, we converge. The RMSD becomes something where actually it's going to be 0 if they're exactly same. But if they may be different length, we may get another RMSD here. Here's another figure from their paper. And the score is actually takes into account, it's based on this distance, the real distance between I and j residues, but there's also certain constants which convert this score, convert the distance to a score. In other words, after the superimposition, when two residues are exactly on top of each other after superimposition, they are very close. They need to get a high score. So a small distance should imply a higher score. So there's an inverse relationship between score and distance. So in order to convert distances to scores, they use this equation, which these are m and d0 are constants. For example, if dij is 0, it means that they're exactly on top of each other. We are going to have here m over 1. So m is the maximum score you get. So m is the maximum score. The parameter m is the maximum score. And d0 is something like it acts like a regularization parameter, I guess, because when dij is infinity, a score can be 0. Okay, I mean, but the d0 is just like a regularization parameter. Now, that's the structural algorithm. I hope that the algorithm is clear. Do you have any questions? Okay, so at the end, it gives us a number of matched residues, and I'm going to stop here. We only have the Dali method left, and actually, we can finish it in like 15 minutes, which gets us back to schedule. Okay, we were behind schedule like 1.5 weeks, but I checked the schedule today. Today we were supposed to finish the structural alignment techniques, and we almost finished them. We only have Dali left, so we are almost back on schedule. Next week we are going to start microarray analysis techniques. Let's check the schedule again quickly.